Ladies and gentlemen, welcome to my YouTube channel, and if this is the first time you are coming, please subscribe to the channel. So today we are going to look at the basically pentose phosphate pathway. So generally when glucose is transported into the cell, it converted into glucose 6-phosphates, and the glucose 6-phosphates will enter into glycolysis to generate acetyl CoA, and the acetyl CoA will enter strict acid cycle followed by the electron transport chain. But there are situations where when glucose 6-phosphate is generated from glycolysis or any other sources, the glucose 6-phosphate will be channeled into the pentose phosphate pathway. But generally, pentose phosphate pathway is not a catabolic process. It's not a catabolic process like in the case of glycolysis. It is usually an anabolic process where some important biomolecules are generated in the body. So pentose phosphate pathways mostly involve in the synthesis of important biomolecules in the body, and it usually occur. It is very, very important in the red blood cell. It is very, very important in the red blood cells. So then the pentose phosphate pathway is also known as hexose monophosphate shunt, and it usually takes place in the cytosol. And generally, the pentose phosphate pathway is mostly active, it mostly active in the liver, mammary gland, and adrenal cortex, and specifically in the cytosols of the cells. And then this pentose phosphate pathway is divided into two. We have oxidative phase, and then we have non-oxidative phase. So what is the purpose of the pentose phosphate pathway? The major purpose of the pentose phosphate pathway is to generate NADPH and ribose 5-phosphates. And the NADH generated is usually important in biosynthetic reactions. An example of biosynthetic reactions in the body includes like steroid biosynthesis, fatty acid biosynthesis, and it is also important in oxidative stress defense. Generally, as a lot of metabolic reactions are taking place in our body, we usually generate oxidants. Example, we have something like hydrogen peroxide, that is H2O2. So the H2SO2 and other oxidants, they are very dangerous in our body, and we have to eliminate them. So this NADPH is important in preventing the oxidative stress in the body by neutralizing the oxidants. And of course, the ribose 5-phosphate is generated, which actually one of the components of the nucleotides that are used in the synthesis of nucleic acids, which include DNA and RNA. So it means that basically in the pentose phosphate pathway, two major biomolecules are generated, which is NADPH and ribose 5-phosphate. So the NADPH is used in biosynthetic reactions such as fatty acid biosynthesis, steroid biosynthesis, and so many other reductive biosynthetic reactions, and also itself in the body in preventing the body from oxidative stress. And then the next one is the ribose 5-phosphate, which generally generates nucleotides, and of course the ribose sugar is one of the components of the nucleotides. And of course the nucleotides are the major building blocks of the nucleic acids, and of course the nucleic acids are DNA and RNA. So now, which molecules control the rate of the pentose phosphate pathway? That is the control point of pentose phosphate pathway. So generally there is one major enzyme, which is the first enzyme of the pathway. Glucose 6-phosphate dehydrogenase is the major control point. It is the rate limiting enzyme. So when it's a control point, it means that it's a point where you can either increase or decrease off or on the pathway. So glucose 6-phosphate is the major enzymes. So now we are going to start with the oxidative phase. So in the oxidative phase, there are three major reactions where first glucose 6-phosphate will be converted into 6-phosphogluconolactone. 6-phosphogluconolactone by the action of an enzyme glucose 6-phosphate dehydrogenase. And in this step, one molecule of NADPH is generated. And this step is like this step is dehydration reaction, is a dehydration reaction. And then the 6-phosphogluconolactone is then converted into 6-phosphogluconates using an enzyme called gluconolactonase. And then finally the 6-phosphogluconate is converted into ribose 5-phosphate. It means that at this stage, the most important thing is that number one, two molecules of NADPH are generated in the first step of the reaction and the last step of the reaction. So the enzymes that involved in the generation of this NADPH are glucose 6-phosphate dehydrogenase and then 6-phosphogluconate dehydrogenase. So at the last step where ribose 5-phosphate is generated, carbon dioxide is also removed. So it means that this is a decarboxylation reaction. And then the next one is the non-oxidative phase. In the non-oxidative phase, the two molecules of ribose 5-phosphates is then the ribose 5-phosphate can be converted into ribose 5-phosphate using ribose 5-phosphate isomerase to generate ribose 5-phosphates. And then of course at the same time, another enzymes can also convert the ribose 5-phosphate through ephemerase, convert ribose 5-phosphates to xylulose 5-phosphates. And then of course an enzyme called transketolase will now condense the two molecules. So in this step, condensation and cleavage are taking place. So at this condensation and cleavage, by the transketolase enzymes, two molecules are generated. Of course, when ribose 5-phosphate and xylulose 5-phosphate are condensed, you expect to have a 10-carbon compound. So then, of course, it will clip at the same time, generate glyceraldehyde 3-phosphates plus celluloheptulose 7-phosphates. So it means that transketolase enzymes are enzymes that catalyze both condensation and the cleavage reaction. And then at the same time, another enzyme called transaldolase is now converts the two molecules, that is the glyceraldehyde 3-phosphate and celluloheptulose 7. It will now convert it into three compound or three products. And this product includes glucose 6-phosphate, erythrosis 4-phosphate, as well as the xylulose 5-phosphate. And then at the same time, of course, glucose 6-phosphate is a substrate for glycolysis, so it can be channeled with glycolysis. And of course, it's a reversible reaction. And then the xylulose 4-phosphate and erythrosis 4-phosphate using another enzyme called using the same enzyme transketolase now generates another molecules of glyceraldehyde 3-phosphate and then fructose 6-phosphate. And of course, the glyceraldehyde 3-phosphate can also be channeled glycolysis, and then fructose 6-phosphate can also be channeled to glycolysis because they are all substrates or biomolecules that are needed for glycolysis. So ladies and gentlemen, briefly, this is actually a concept of pentose phosphate pathway. Please, if this is the first time you are coming to my YouTube channel, please subscribe. Thank you.